Hello, I'm Eloise Keefe, the Business Manager for the Institute for Glycomics at Griffith University in Australia. I am delighted to share with you a brief snapshot of the Institute during BioCareer's Biotech Playground and look forward to deeper discussion during the online partnering and other activities this week. Established in 2000 through investment by Griffith University and the Queensland Government, the Institute for Glycomics is one of Australia's flagship interdisciplinary translational biomedical research institutes, based in the heart of Griffith University's Gold Coast campus and the Gold Coast Health and Knowledge Precinct. The Institute boasts state-of-the-art facilities combined with some of the world's most outstanding researchers focused on glycomics, a constantly expanding field that explores the structural and functional properties of carbohydrates, or sugars, and their roles in disease. Our research engages worldwide partnerships in projects that cut across multiple disciplines to apply new approaches to the identification, treatment, and prevention of disease. And together, it is the vision of over 200 scientists and support staff to establish the world's preeminent Glycomics Research Institute for the discovery, development, and commercialization of novel drugs, vaccines, and diagnostics for infectious diseases and cancer. To achieve this, we maintain a diverse portfolio of drugs, vaccines, diagnostics, and research platforms. The Institute has a strong focus on commercialisation of our technologies, and we seek the best partners, both locally and globally. In our experience, there is great synergy to be had when partnering with Asian biotech and pharma, which is highlighted in the number of recent deals closed with Asian partners. By combining the Institute's expertise in discovery research through Phase 1 clinical trials with company partners' expertise in late-stage product development, marketing and distribution, we can take advantage and provide a smooth pathway for each technology through to market. This is further enhanced by our typical deal structuring, which encourages commercially focused technology development and excellent technology transfer to industry. Today, I'll briefly highlight 3 technologies that are available for partnering within the Institute. Please do reach out if you would like to discuss these or other aspects of the Institute for Glycomics further. The first technology is a vaccine candidate for the prevention of Group A streptococcus, which is responsible for about 800 million cases of well-known but relatively harmless diseases such as pharyngitis and impetigo each year. However, at least 18 million of these cases progress to much more severe strep A diseases such as rheumatic heart disease and streptococcal toxic shock syndrome. With no vaccines currently available to prevent the disease, Professor Michael Good and Dr. Manisha Pandey are meeting an urgent unmet medical need with their peptide-based vaccine candidate, which targets a conserved region of the M protein and confers broad protection across multiple strep strains. The vaccine candidate will enter a Phase 1 dose-ranging study in Canada this month and is expected to start a Phase 1b challenge study in Australia by the end of this year. This technology is partnered in China, but available for licensing in the rest of the world. Following on from the previous technology, the same team have developed an immunotherapeutic for the treatment of invasive streptococcal disease and streptococcal toxic shock syndrome. Strep toxic shock presents in around 20% of invasive strep cases and has very high mortality rates, exceeding 40% even in well-equipped hospital settings. The current treatment for strep toxic shock is IVIG. However, studies show that this approach doesn't improve patient survival. Alternatively, our data has shown that an immunotherapeutic for the targeted treatment of strep A bacteria and the exotoxins that they produce shows efficacy in mice. Development of this technology will finalise the production and humanisation of monoclonal antibodies and support completion of a Phase 1 clinical trial in Australia to evaluate safety. The final technology to present today is a therapeutic to potentially treat and prevent gonorrhea in females. Gonorrhea is the second most commonly reported sexually transmitted infection, affecting over 106 million people each year. Alarming increases in incidence over the past 5 years has earned gonorrhoea status as an urgent public health threat by both the Centers for Disease Control and Prevention, the CDC, and the World Health Organization. Neisseria gonorrhoea, the bacterium responsible for gonorrhoea infection, is highly variable and exquisitely host adapted. Its advanced treatment evasion strategies has seen resistance develop against every antibacterial ever used to treat it. Similarly, no vaccine is available despite decades of extensive research. Professor Michael Jennings and his collaborators at the Abigail Wexner Research Institute at Nationwide Children's Hospital in the US have discovered that methyl-dopa, a generic drug used to treat hypertension in pregnant women, therefore very safe, may both treat and prevent gonorrhea infection in women. These data have been obtained using ex vivo models of human primary cervical epithelial cells. Due to an absence of suitable animal models, we're preparing to take advantage of Australia's favourable regulatory system and enter a phase 2 efficacy study in the coming months. We hold a patent on this new use of methyl-dopa. As I mentioned earlier, please do get in touch if you wish to discuss these technologies or others in our pipeline for either commercial or research partnerships. I look forward to speaking with you.